replication once inside a host cell. With all of these parts, it makes sense that human herpes viruses are on the larger side as far as viruses go, typically spanning about 150 nanometers in diameter. However, like other viruses that contain an envelope, human herpes viruses are sensitive to solvents, acid, detergents, and drying. Now that we've familiarized ourselves with human herpes viruses in general, we can get more specific. Herpes simplex viruses, or HSV, were the first in their group to be recognized. The name herpes comes from a Greek word meaning to creep. There are two types of herpes simplex viruses, HSV-1 and HSV-2. They share very similar characteristics, like DNA homology, antigenic features, symptoms, and tissue tropisms. However, there are some subtle but significant differences that set them apart. Let's think about the genetic makeup of HSVs in particular. The double-stranded DNA genome of HSVs encodes somewhere around 80 proteins. About half of these are involved with creating more copies of itself in the way of replication, meaning things like a DNA-dependent DNA polymerase. The other half control how the virus interacts with the host cells and the host's immune response, so at least 10 glycoproteins that serve as viral attachment proteins. Unfortunately for us, HSV can infect most types of human cells, and even cells of other species. However, we should note that although HSV can infect other animal cells, HSV infection is exclusively a human disease. Once the virus has attached to a cell, it penetrates the host cell by fusing its envelope with the cell surface membrane. Once the virus has fused with the host cell, it releases its capsid into the cytoplasm of the host cell, along with some enzymes that help the virus make more copies of itself. Now, HSV has evolved a very sophisticated and stepwise system to replicate itself. The first things created are what we call the immediate early gene products. This includes DNA-binding proteins, which help stimulate DNA synthesis and transcription. Next up are the early proteins, which include catalytic proteins like DNA-dependent DNA polymerase to kickstart DNA replication. Replication of the viral genome then triggers transcription of genes that encode structural proteins and other necessary proteins for viral function. From here, the virus is assembled piece by piece before being released either by exocytosis or cell lysis. In the case of neurons, when compared to other types of infected cells, the virus may either replicate immediately or establish latency, depending on which viral genes are expressed.